The cell-mediated response, the topic of this video, is one of the two different responses that we attribute with the third line of defense as a component of the adaptive or specific form of the immune system. The goal of this video is to cover concepts associated with how the cells, the chemicals, work together to coordinate a very complex but highly effective response. So let's get started. There are a handful of key players that you need to be familiar with to further understand or to better understand the third line of defense's cell-mediated response. So the cells that we'll be investigating during this video include the T helper cell. We're going to denote that as a T with a subscript H. We're also going to take a look at the cytotoxic T cell. We're going to denote that as a T with a lowercase c. So these are our cells. And we're also going to investigate how B cells can also be coordinated and stimulated to respond when the cell-mediated response is occurring. Now, we also have a number of chemicals. And those chemicals are going to be a key component in helping coordinate the activity of these three different cell types over here. The key chemical we're going to look at, we're broadly going to refer to as an interleukin. Now, depending upon your course, not in mine, but in some others, they might expect you to know that a cytotoxic T cell and a helper T cell have alternative names. So a cytotoxic T cell is sometimes referred to as the CD8 cell, and the T helper cell is sometimes referred to as the CD4 cell. In this video, though, we're going to stick with the language helper and cytotoxic T cells. B cells also have a special naming system. The interleukins then that are produced are the communication piece between these different cells. And we won't refer to the individual interleukins, interleukins 1, interleukin 4, interleukin 8, etc. But I will joke through this video about the language of the cells. And those are really those interleukins. We'll talk a little bit more about those. Now, if you watched the third line of defense video that I have published, you will note that the cytotoxic T cells are also producing a chemical, another substance that's being made, and those chemicals are the perforin, kind of sounds like perforate because that's what it does, and granzymes. And if you watched that third line of defense video that sort of summarizes the anatomy of the third line of defense, you will have also known then that B cells produce something. It's a protein known as an antibody. So these are our key players, and we're going to kind of get rolling here to see how these chemicals and cells work together to make the cell-mediated response. Wow, this is a lot. But let's zoom in and investigate this process bit by bit by bit. We're going to start over here in the upper left-hand corner looking at the macrophage and a naive helper T cell. Note the number 1. The macrophage will have previously digested foreign particles, and if it determines that there is something that is evil, dangerous, doesn't belong, it's going to present that antigen on a signaling protein we know as the major histocompatibility complex 2, or MHC2. So each of the macrophages in this image show their MHC2 with a red ball antigen. Inside a lymph node, which is a great location to bring immune system cells together, as well as bringing microbes and pathogens together, in this lymph node, there are going to be a number of T helper cells. These T helper cells were created in the bone marrow. They went to Thymus University. They went to the thymus gland and learned about self versus not self and dangerous versus not dangerous. And even still, we consider them naive. And we consider them naive because they really don't have background knowledge against foreign cells. So a naive T cell in a lymph node will interact with a macrophage. And the macrophage is going to release chemical signals we call interleukins. Now, I like to joke that this is a very specific language called macrophages, because this level of signaling, this type of signaling, is a special signal that goes just between a T helper cell and between a macrophage. As these interleukins are moving back and forth, this chemical signaling, the T helper cell is also touching the red ball antigen. And as it's touching the red ball antigen and receiving these interleukins, it's receiving a message that says, hey, we have a wanted poster for this red ball antigen. We need to go out and seek to destroy it. Now, a T helper cell is going to be savvy because the T helper cell has the ability to start a very intense and very large response, as you saw in the initial screen here. So, a T helper cell is going to double check, maybe triple check, to make sure that what is being asked of it to mount a response against this foreign antigen, the red ball antigen, is this really something that needs to happen? So a T helper cell will interact with other macrophages, looking for the major histocompatibility marker 2 with the red ball antigen, and exchanging interleukins between multiple macrophages and the T helper cell to truly determine whether or not something is dangerous and whether or not we need the third line of defense, the cell mediated response, to engage. So we call this stimulation between multiple cells co-stimulation. And once a T helper cell has been co-stimulated, that T helper cell is going to be carrying the knowledge about the evil red ball antigen. So coming over here to step 3, we see we have a T helper cell with a red ball antigen. And now that cell is going to go through a process known as clonal selection. Now this is really just fancy talk for mitosis, which is the creation of identical daughter cells, but this is a little bit more than just mitosis because every daughter T helper cell that is created will have knowledge about this evil red ball antigen. And the rate of mitosis is going to take a little bit of time, so typically with the first exposure to a new antigen, to something that a macrophage is presenting as dangerous, it will take the cell-mediated response on average 7 to 14 days to develop a cohort of cells and be able to have a true effect. Once clonal selection of T helper cells is completed, we now have a whole clone army, throwback to the Star Wars movies, of T helper cells. But T helper cells are more like a general. They're more like an individual that's going to coordinate other cells rather than do any of the dirty work themselves. So we're going to move over here from 4 in clonal selection. We're going to take a peek at what a T helper cell with knowledge of the red ball antigen can do. We're going to start over here with 5A. And 5A shows a T helper cell bumping up against and communicating with a naive cytotoxic T cell. Now, cytotoxic T cells do like to get their hands dirty. They are a cell that gets right out there and starts destroying. I sometimes think of these as the Navy SEALs. But a naive cytotoxic T cell is essentially like a cell or a person going into boot camp to become the Navy SEAL. So cytotoxic T cells are sometimes referred to as effector T cells. The helper T cell in 5A is going to bump up against a cytotoxic T cell. This shouldn't be too difficult if everybody's in the same lymph node. And when these cells interact, a different form of interleukin is released. The form of interleukin released here, I joke, is cytotoxins, but this T helper cell has to be able to communicate in a very specific way with a very specific group of interleukins to communicate to the naive cytotoxic T cell that there is an enemy that is in the body that needs to be looked for. Just like we saw in the helper T cell, 6A, step 6A, is going to be that cytotoxic T cell now aware of the enemy, the red ball antigen. This is the target now for this cell. And the cytotoxic T cells then begin their own steps of clonal selection. And that process of clonal selection is once again going to create an entire clone army of cytotoxic T cells, not just normal mitosis though, because in this clonal selection process, each of these cells is aware of the red ball antigen and knows that it needs to be able to attack. From there, those clonally selected increased numbers of cytotoxic T cells with knowledge about the red ball antigen are going to then go into step 7, the seek and destroy phase. Remember, these are sometimes referred to as the effector T cells because they get their hands dirty. They're the ones that are actually out there in the field creating the desired effect. So 7A, the cytotoxic T cell is going to seek out foreign cells. And when it finds that foreign cell with that red ball antigen, this time not on an MHC2 marker, but on a separate marker, the cytotoxic T cell is going to be able to recognize that as the pathogen that doesn't exist. Now, cytotoxic T cells, knowing that they're going to war, come heavily armored. And so in vesicles, they have pre-created digestive enzymes known as perforins and granzymes. And when the cytotoxic T cell identifies a cell that needs to be attacked, they then release those perforin and granzymes to attack the cell. And that's going to poke holes in the cell that's going to help destroy the microbe or the foreign cell that the cytotoxic T cell has been told to look for. So 8A is the destroy component of the cell, and you can see with the X here, right, we wipe out the cell once we release those digestive enzymes. What's absolutely amazing about this system is all the while a couple of T helper cells are coordinating with the cytotoxic T cells to mount a response. At the same time, a different group of T helper cells can be working with B cells. Now, B cells are a part of an immune system response that we call the humoral immune response. But in this case, the humoral response is dependent on a T cell. And so that helper T cell, a different group of helper T cells, says, okay, well, you guys go over there and communicate with cytotoxic T cells. A different group of helper T cells identifies B cells that are naive, that need to be educated. Once again, interleukin is going to be exchanged between a T helper cell and a B cell, but this time it's a different interleukin. It's a special language spoken just between the T helper cells and the B cells. I like to think of the T helper cells as being a multilingual cell. And so in this case, they're speaking B cellese, and the naive B cell, which this B cell could self-stimulate. The B cell itself can pick up the red ball antigen and ask itself, what do I do with this? Should I attack this thing? Is this something that is truly dangerous to the body? So that's our 5Bs. This is 5B because simultaneously 5A is happening. While some helper T cells are activating the cytotoxic T cells, another group of helper T cells are activating the B cells and triggering the humoral response, the dependent humoral response. Once the B cell has been educated, the confirmed enemy of the red ball antigen, clonal selection happens right all over again. More mitosis, more B cells, all that have the knowledge of that red ball antigen. And once they have that knowledge of that red ball antigen, now they are going to get to work. And their role is slightly different. So these are the cells, maybe you might think of them as the cells that are making ammunition to be able to attack. So a group of B cells that have gone through clonal selection are going to differentiate. They're going to change shape and become known as plasma cells. Now, plasma is kind of a throwback to the fluid component of the blood because the goal of the plasma cell is to increase the rate of transcription and translation, which produces antibodies. So first, 7B, B cells that have undergone clonal selection are going to start to differentiate into plasma cells. And then plasma cells are going to increase their rate of transcription and translation. That's what the T-T stands for on the board. And they're going to crank out antibodies. And the Y shape of the antibody is what we consider the constant end. That is the general pattern of all antibodies. However, there's a variable end at the tip of each of the Y. And so you'll notice here that they're illustrated in a half moon shape so that they can bind to the ball antigen. Once the plasma cells have cranked out antibodies, those antibodies then can bind to those antigens that they were made for. So over here, back to our predatory, evil microbe. You can see an antibody with both of its variable ends, those roundish ends, cupped and attached to the antigen, and now the antibody is starting to coat this microbe. There are a lot of functions to antibodies, which include binding antigens together, coding antigens for neutralization. Antibodies are a key player in this process. But here's the limitation. Antibodies as a protein last approximately 6 months. Then we got to make more. But the nifty thing about the cytotoxic T cells is that all of our T cells are going to be long-lived cells. They have a huge longevity. And so these cytotoxic T cells are going to last years and be in circulation for years. Now, I wish I could tell you that that was it. But one of the key aspects of the third line of defense is to have immunity and to have memory to these foreign antigens to be able to mount a response in a much more rapid frame of mind, a much more rapid state of existence. So if we look here to the center of the screen, you're going to see a little arrow that says memory. And while a whole cohort of T helper cells are activating the third line of defense cytotoxic T cells, and another group of T helper cells are activating the B cells and engaging the B cells, a cohort of each of these types of cells is immediately going to be put on hold. A cohort of all three of these cells is going to be created to be like the National Guard, fully trained, but not called to action. So what we find in the human body is that some helper T cells become memory helper T cells. A cohort of the cytotoxic T cells become memory cytotoxic T cells, and even the B cells, a group of B cells, are going to become memory B cells. The benefit to having all of these memory cells is that the clonal selection has already happened. The mitosis rate has already happened. So, if the body is infected with the evil red ball antigen or whatever disease, whatever microbe we're talking about, if we get a second exposure or a third exposure or a fourth exposure, these memory cells are able to respond much more rapidly. So you see the little note here that says second exposure, you can get a response in 24 to 48 hours. That happens much, much faster than this whole process of T helper cells learning information from a macrophage with their interleukins, clonally selecting, communicating with a different set of interleukins to the cytotoxic T cells, and those cytotoxic T cells going through clonal selection, and helper T cells activating the dependent humoral response, getting B cells to start their activity, clonally selecting, making plasma cells, creating antibodies. This memory component allows for a much more rapid response if that antigen, the red ball antigen, is ever seen again. Time to summarize the cell mediated response, CMR, cell mediated response. We looked in this video at T helper cells and their direct ability to interact with three other cell types. They interacted with the macrophage. That's where they got the information about the foreign cell that needed to be destroyed. The T helper cell also communicated with the cytotoxic T cell, able to coordinate and educate the cytotoxic T cell on what to search and destroy. And we looked at the cytotoxic T cell producing two chemicals, those chemicals being perforin and granzymes. These were things that these cells could use directly to affect, target, and attack foreign cells, any evil red ball antigens or any other antigen that the T helper cell learned from the macrophage was indeed foreign. The T helper cell also was helpful in activating B cells and creating what we call the dependent B cell response. And when the B cells respond favorably to the helper T cells and their signaling, we get the production of antibodies. Now, from all of these, we get a whole cohort at the end of this process of memory cells. And those memory cells are really important to us because these memory cells can respond quicker. They typically can start to mount a response in as little as 24 to 48 hours. However, if we think about the cell-mediated response and all of the clonal selection and all of the signaling and all the things that are going on inside a lymph node, inside an infected tissue, one of the things to note is that this, during that first exposure, is timely. And on average, it takes 7 to 14 days after that first exposure to get this full cell-mediated response up and running. So, we therapeutically would utilize something like a vaccine potentially to get this first exposure over and help create a cohort of memory cells that is going to help a patient be able to fight off an infection, ideally without ever getting sick. But if the patient gets sick, and patients who get vaccines will get sick, perhaps we get a quicker response of these cells within 24 to 48 hours. And so another goal of vaccines is to reduce the severity of the disease. And the cell-mediated response is going to be able to help this. Hopefully you were able to see that with the cell-mediated response, this is a system that is going to engage and be highly effective because the cell-mediated response is able to engage the humoral response while also activating cytotoxic T cells to be able to destroy. Important to note that the humoral response can self-activate in a process known as the independent humoral response. And to take a look at how that's happening, consider watching that video. I hope this helps you better understand all of the nuances of what the cell-mediated response is doing in order to coordinate an attack and be able to create long-term immunity or memory to that specific foreign molecule. Have a great day.